Hello. Chapter 3, it's about cell biology. In this chapter, we're going to learn the different characteristics of the cell, the structure of the cell, the functions of the cell, and as well, how the cell divides. So the cells have several functions, and one of the functions is to perform metabolism and use energy. And the cells also are important to synthesize molecules like proteins, lipids, and nucleic acids. The cells as well communicate with other cells and they can receive or send electrical and chemical signals. The cells as well can reproduce in order to repair damaged cells or to make some of the tissues to grow. And the cells have within their nucleus, which is one of the parts of the cell, the organelles, they have DNA that it is important for the transmission of these genes, for instance, in meiosis, which is important for heredity. So some of the major structures of the cells are a plasma membrane, which forms like a wall or a separation between the cells and other cells. The nucleus, it is the central part of the cell where the DNA or the nucleic acid directs the activities of the cell. The cytoplasm is the space between the plasma membrane and the nucleus, and it is where the tiny organs or organelles are located. So this is an example of an animal cell or a human cell. And in this cell, we have here the nucleus. The nucleus is surrounded by this nuclear envelope, and inside it has this fluid that we call the nucleoplasm. And it is in the nucleus where you have this organelle that is called the nucleolus. And the nucleus is surrounded by this other organelle that is called the rough endoplasmic reticulum. And this cell has as well the powerhouse of the cells, which are the mitochondria, and an organelle that packs and distributes the proteins, which is the Golgi apparatus, along with proteins that makes the cells to have their specific shape into what we call the cytoskeleton. And the cells also has different vacuoles like lysosomes, proteasomes, that helps in the recycling of materials for the cells, or in the case of the cells, when they are worn out, it helps for the cells to be recycled. And then the cells also have a part of little organelles that are called the centrioles that help to separate the chromosomes that are found in the metaphase cycle of mitosis or stage of mitosis when the cells are about to divide. Now, this is the plasma membrane, the boundary that separates these cells from other cells, and within this plasma membrane, these cells may have extensions like cilia, which are long extensions that are used for the cells to sweep away materials, or microvilli that it can be used for absorbing nutrients. Now the cells as well, they have in their cytoplasm free-floating ribosomes that are important to make proteins. The cells are studied in cytology, which is the study of the cells, with the aid of microscopes. The cells are generally too small to be seen with the naked eye, so we need to use microscopes. Now, some microscopes, like the ones that we use in the classroom in biology intro classes, are what we consider compound or light microscope. This has a magnification of the specimens up to 1,000 times their size. And in the case of the cells, it helps us to know their structure and their organization and their type. For instance, we can study biopsies of tissue, which is living tissue, to see the characteristics of that tissue. Most of the cells cannot be seen in light microscopy without the aid of a dye or a stain, so most of cells, they need to be stained. Another types of microscopes that we have available, but they require a highly specialized training to use them, is a scanning electron microscope, or SEM, and transmission electron microscope, or TEM, that sometimes can magnify the images up to 100,000 times. And then we have atomic force microscope, or AFM, which is made out of sophisticated components, including a probe that can go and scan the internal parts of the cell, and it reveals up to the details of DNA, ribosomes, etc. So this is an example of some images from microscopy. In this case, we have this cell that has been seen with what we call actually fluorescence microscopy. In this case, you dye the cells with special fluorochromes or dyes that emit at a higher level the light that they absorb with these fluorochromes, and you can see better the details. So this is the plasma membrane of the cell, and this is the nucleus. And this is an image with an electron microscope. So this is the image of a very detailed structure inside the cell. And this is the image of pores within the cells. And this is the image of three-dimensional view of these pores within the cells. So these are pretty much images from very sophisticated microscopes. Now we need to study the individual components of the cells. One of the individual components is the plasma membrane. Function of the plasma membrane is to separate the internal materials of the cells from the external environment. And this plasma membrane, it is made of phospholipids, and these phospholipids have these fatty acids that faces towards the portion where you don't have water inside and outside of the cell. So we have two phospholipid layers that we know as phospholipid bilayer. And then you have the fatty acids then that face internally, and they have as well a phosphate group that it is polar, that is charged, that it can interact with water. So these phosphate groups can interact with the water outside of the cells and inside of the cells. Now the plasma membrane has embedded carbohydrates, lipids, and proteins that have a specific shape and size and function. So we will see all of those. And the phospholipids of the plasma membrane and some of the components, including cholesterol, they can move around. And this is what we call the fluid mosaic layer. Now, the plasma membrane will play a role in certain cells, like excitable cells, to perform an event that it is called the action potential that helps for the cells to function. So if you magnify this part of the cell from the previous model that we saw before, and you see the components of the plasma membrane, so here it is. This is the phosphate groups or heads from these lipid layers, and these little yellow lines that you see, these will represent the fatty acids. So there are two fatty acid tails attached to a phosphate group. And this is what we consider the external membrane surface. The internal membrane surface is similar. It has a phospholipid layer, and both of them, if you see here, are called a phospholipid bilayer. Embedded within this phospholipid bilayer, you have these proteins. Some of them allow the entry of molecules through the cells. So these are channels. These are what we call membrane channels. And some of them, they don't have an opening. And they can be embedded only in the external membrane surface, and these you call them integral proteins. Now, other integral proteins, they can have attached carbohydrates into these proteins. And these proteins will help to sense signals from the environment. So these are receptor proteins. And some of these proteins as well will have connected these long carbohydrate chains, while some of them, they may be branched, like in this case. And then you can have as well integral proteins that attach into the internal membrane. And some of these proteins, or even these internal membrane surfaces, can be attached into these proteins that are found within the cytoplasm of the cell that we call the cytoskeleton. Now, within this plasma membrane, you also have these molecules of cholesterol that helps for these phospholipids to be a little bit less flexible so that you don't have a flimsy plasma membrane. So, characteristics of the membrane lipids. So, phospholipids and cholesterol predominate the membrane lipids. And again, the phospholipids have a polar head that faces the interior and exterior of the cell where you have water. And because they're polar, they can interact with water. So they are called hydrophilic polar heads. And they are made of phosphate groups. The nonpolar tails, they face each other in the interior membrane and they are hydrophobic. And then the cholesterol are interspersed within the phospholipids to make a little bit more rigid this plasma membrane. Characteristics of the membrane proteins. So the integral membrane protein can extend deeply into the membrane, or they can extend from one surface into the other. And some of them, like in the case of protein channels, they have an opening. Now we can have peripheral membrane proteins that are attached either to the inner or outer surface, and these will have different functioning depending on the three-dimensional shape and the chemical characteristics. So some of these peripheral proteins can be markers. Other ones, they can adhere the cells to one another, so it's what we consider the adhesion molecules. Other ones can be channels, receptors, enzymes, or carriers. So in the case of the transport proteins, so the transport proteins are channel proteins. And they have the hydrophilic region, the one that faces inward, located here, while the other part will be facing outward. And in order for these molecules that you see here, which are represented by these pink spheres, to pass into this channel protein, you have a characteristic shape and electrical charge. Otherwise, these channels do not allow the passage of particular substances or molecules. Now, some of these proteins will be channel proteins, but other ones will be carrier proteins, like in this case. In this case, this protein, which is a carrier, will physically interact with this molecule, and then it will help to carry it or bring it into the cell. And other proteins function to pump molecules by using energy, by using ATP. Channel proteins, they can be classified as leak ion channels or gated ion channels. Leak ion channels will be always open, and they have a specificity. So we have leaky channels, for instance, for sodium, and only sodium can cross by these leaky channels. And we can have gated ion channels that they have a gate that can be open or closed depending on what stimuli they receive. And gated ion channels can be subclassified as ligand gated or voltage gated. Ligand gated will be those ones that require the physical connection of a molecule so that they can open. And voltage gated, they need to have these stimuli that it is an electrical stimuli that can increase the gap between these gates so that you can open these channels and then you can allow the crossing of molecules. So voltage gated channels respond to changes in electrical current, while ligand gated responds to the physical connection of molecules to specific areas within these channels. So this is the diagram to show you the different type of channels. So this is a leaky channel for potassium, so it's always open, so potassium can move freely. And then in this case, we have a sodium ligand gated channel that it is closed. And when you have a molecule, like for instance acetylcholine, binding into this area of the gate, the gap between the gate, like in this case when it was closed, it was non-existent, but opens, and then you can allow the entry of these molecules of sodium. Carrier proteins are also known as transporters. They are located in between the inner and the external layers of the plasma membrane, so they are integral proteins, and they have specific binding sites for specific molecules. So not every molecule can be transported by the carrier proteins, only the ones that are specific. But once this molecule binds and it has the specific shape, then there is this change in the shape to carry the molecule. So for instance, in here it was open, then the molecule enters, and then in this mid portion of the channel, it connects, and then the channel closes here exteriorly and opens interiorly to allow this molecule to be transported. Now, carrier proteins can be subclassified as uniporters if they only move an ion or a molecule, or they can be symporters if they carry two molecules in the same direction at the same time, or they can be antiporters if they move two molecules or two ions in opposite directions at the same time. For ATP power pumps, they require ATP, and they use the energy to allow the cells to accumulate substances. The rate of the transport depends on how much molecules you have available to transport and how much ATP. So if you don't have so many molecules to transport, you don't transport as many. And if you don't have ATP, the energy, you don't transport these molecules. So here we have an ATP power pump. So in here we have this ion moving into the outside of the cell, and you have attached a molecule of ATP into this side of this pump. And as this ion is transported, this ATP will be converted into ADP and inorganic phosphate. And then you have extruded then your ion. Receptor proteins are proteins like glycoproteins or just proteins without the glucose or the carbohydrates. And these are located in membranes. that has an exposed receptor site. So these can attach specific molecules, and then they can help the cell to detect something that is going on in the environment. And these molecules that attach to these receptor proteins are known as ligand, and these ligands are specific for these receptors. Now, some receptors have molecules that they have to connect into a specific area. Like in this case, you have these receptor sites for acetylcholine. So when acetylcholine, which is a neurotransmitter, binds into this area, then you open the gate for sodium, and then sodium will enter into the cells. So you change the permeability or the substances, how many they will enter into the cell by having this type of proteins. Some proteins internally have these molecules that can interact with. So this is what we call the G protein complex. This G protein complex will interact with this molecule. So by being close by, it will have the ability to just move forward until it contacts this molecule or this receptor, and then this receptor will make a change. It will allow the breaking down of one of these units, which is called the alpha unit, from this molecule, and then this molecule is going to stimulate a cell response. Enzymes are catalysts. They can increase the speed of chemical reactions, and some of these enzymes are located within the plasma membrane. And when these enzymes, for instance, in the case of these enzymes that are located within the intestines, bind proteins like the dipeptides, these enzymes can break down these dipeptides into amino acids so that these amino acids can be absorbed by the intestinal cells, and then we can perform metabolic reactions with them. Now, the plasma membrane, it is not completely sealed. It is having then these channel proteins that allow the passage of substances. But since not all the substances can cross through these channels, then this plasma membrane is selectively permeable, meaning that it will allow only certain substances to pass through it. And the substances that can pass through this plasma membrane can be passed through the cells by different transport mechanisms, either by passive transport, in which you don't spend any energy or any ATP, and this passive transport can be subdivided into diffusion, osmosis, or facilitated diffusion. Other mode of transport is known as active transport. In this case, you utilize ATP, and vesicular transport is when the cells bring in materials into what we call endocytosis, or they can export materials into what we call exocytosis. Some details about diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. The greater the difference between the concentration, the much faster the diffusion will happen. So, then molecules will move from more to less areas. And this difference between the concentration is known as concentration gradient. Now, diffusion also will be influenced not only by how many molecules you have in one area versus the other one, but also it can be affected by the size of the molecule and also the temperature and also the permeability of that particular membrane. In the case of temperature, when you increase temperature, remember, you increase the motion of the molecules, and this makes the molecules to collide, and with this collision, you can move the molecules much faster. So you can make them to diffuse faster. So that's for temperature. Now, if you have a membrane that only has 10 channels, this membrane might not be as good to diffuse molecules as one membrane that has 1,000 channels. So if the plasma membrane has more channels for a particular molecule, this molecule will get transferred much easier. Another thing that plays a role in diffusion is the size of the molecule. The bigger and the heavier the molecule it is, the harder it will be to move. So smaller molecules can diffuse faster than bigger molecules. Now, viscosity is a physical property of certain fluids to resist to move or to flow. So if you have a viscous fluid, the movement wouldn't happen as easy as a fluid that it is less fluid, less viscous. So this is an example to make the point that diffusion happens for more concentration to low concentration. So in this case, you have a beaker filled with water. And when you add a salt crystal with green dye in the bottom of this container or this beaker, the molecules will start diffuse from the bottom of the beaker into the mid part and then ultimately toward the entire water that is filling this beaker until you have dissolved this salt. And this is the movement then of molecules. So from higher concentration on the bottom to lower concentration at the top until you reach equilibrium in this case. And you can see all these beaker filled with water with green color. Now then, this is the example of diffusion within the plasma membrane. So let's say that you have here 2, 4, 6, 8 molecules that can pass only through this channel. And in here you have 3 molecules of this same type. So since you have here 8, and then in here you have 3, so you have 5 more in here. So you have a concentration gradient, 8 versus 3. So the molecules will move from where you have more or diffuse from where you have more to where you have less. So from 8 to 3. And then some molecules like this non-lipid molecule, non-lipid soluble molecule cannot enter either through this channel because it's not specific or through the plasma membrane. So it will not penetrate. So this is the membrane being selective against this molecule. Now, for lipid-soluble molecules like CO2 or oxygen or steroid molecules or lipid-based molecules, they can cross directly through the plasma membrane without the aid of channel. So this is a case of movement of solid molecules or gases. But what about liquids like water? So water can move as well, and the movement of water through a selectively permeable membrane is known as osmosis. So water will move from an area of low concentration of solutes to an area of of high concentration of solute through water channels that we know as aquaporins. And another way to look at osmosis is basically the movement of water from an area of high concentration of water to low concentration of water. Now, osmotic pressure will be this force that it will be required to prevent water from moving across a membrane by osmosis when you have reached a specific pressure. So this is the example of osmosis. So in here you have a beaker, and then this beaker, it has certain volume of water in it. Now, when you introduce this tube with molecules inside solutes that are highly concentrated and this selectively permeable membrane, and this solution, let's say, is 3% salt solution or saline solution 3%, and you put it here into this beaker with distilled water, and you wait for a certain amount of time, then water will enter into this tube, and then you will have rising of the salt solution. Initially, you didn't have huge amount of salt solution in here up to this level, but the water will be lost from the beaker and enter into this tube until you reach a specific amount within this tube, and this is going to create pressure, preventing more flow of water. So this will be osmotic pressure. Now, these concepts are important to understand because once you have cells placed in different types of solutions, you can have different responses of this cell. So, for instance, if you put the cells in the normal solution where they usually are, if you don't change the number of solutes into what we call isotonic solution, the cells will not gain nor will lose volume, water. Now, if you place these cells in a solution that has high concentration of solutes, which we know as hypertonic, the cells will dehydrate. They will lose water. Because remember, water will move from an area where you have low concentration of solutes to high concentration of solutes, in which case the low concentration of solutes was the cytoplasm of the cells, and the high concentration solute will be the hypertonic solution outside the cells. So then the cells, in order to dilute these solutes, it will lose water. So the cell will shrink or crenate. Now the opposite is true. So when you have a cell and you put it in a hypotonic solution, in which this hypotonic solution doesn't have as many solutes, then the cell will swell. So in this case, we have red blood cells. You can put it in hypotonic solution, and then the cells will swell and eventually will burst into what we call lysis. Or if you put it in a hypertonic solution in which you have high concentration of solutes outside, the cells will dehydrate or crenate. Now, if you put the cells in isotonic solution, the cells will neither gain volume, neither they lose volume. So they will stay the same. So this is how these red blood cells look like. So this is a normal red blood cell in isotonic solution, a swollen red blood cell in hypotonic solution, and a shrink or a crenated cell in hypertonic solution. Now, the other form of transport that we have is facilitated diffusion. You will still move molecules, but in this case, the molecules, they have to be transported by a carrier. And since these carriers, they have a specific three-dimensional shape, these proteins, only the molecule that has that complementary shape can be transported through these channels. And in facilitated diffusion, you don't require energy or ATP. So this example will be for glucose. So we have glucose transporters, and molecules of glucose are very big, but they can enter through these channels, bind to these channels or be transported, and then they will be carried into the cells. Now we have this other form of transport that is known as active transport. Active transport requires ATP, and if a cell runs out of ATP, you don't will transport these molecules, so the transport will be disrupted. Now, in active transport, you can move the substances against their concentration gradient or from low concentration to high concentration. It's like when you have an elevator, and this elevator is full of people already. But if you want one or two more people to enter into the elevator, what you need to do? You need to spend energy. So you got push against resistance. So in this case, active transport can force molecules to move from an area of low concentration to high concentration. And in order for that to happen, you have to spend ATP or energy. Classical example of active transport is the ATP ATPase pump the sodium potassium pump that requires ATP and that helps to create what we call electrical potentials across the membranes. Now, this example that we have here is just to show you what is a concentration gradient. So a concentration gradient, it will be the difference in amount of a substance, a gas, pressure, solute, etc. between two compartments. So for instance, outside the cells, the extracellular compartment, it will have a concentration of sodium of 140 milliequivalents, which is a unit that we use in physiology, per liter. And the intracellular sodium concentration, or the concentration of sodium inside the cells, is minus 12 milliequivalents per liter. So if you subtract the intracellular sodium concentration is not minus 12, it's 12 milliequivalents per liter. So if you subtract 12 from 140, you have a difference or a concentration gradient of 128 milliequivalents per liter. So that's concentration gradient, the difference between compartments of a molecule, a substance, gas, pressure, etc. So again, active transport requires ATP, and it can be used to transport ions like sodium, and it will move the ions back into the cell to provide the energy to move these molecules in different directions. So here is the two examples for active transport. So you have here a sodium potassium pump. So 3 sodiums will being extruded or exported from the cells while 2 potassiums are being brought in. So that's why it's called sodium potassium pump. And since these two molecules are carried in opposite directions, you will maintain a concentration gradient or difference. So you have more sodium located outside the cells and more potassium inside the cells. Now this sodium can be brought back in to the cells by using this cotransporter or symporter that we know as sodium glucose cotransporter. So in this case, sodium will bind and add the same glucose, and glucose can be used to generate more ATP, which is going to make this sodium potassium pump to function. While it will also increase the transport of sodium, and sodium will be extruded by this sodium potassium pump. Another type of transport that happens in the cells is vesicular transport. Vesicles are pouches or vacuoles that can be moving substances within the cells. Now, we have two types of vesicular transport. We have endocytosis, which is moving molecules into the cells or particles. So endocytosis can be subdivided into phagocytosis if you move a solid particle, or pinocytosis if you move a fluid vesicle or a fluid into the cells. And exocytosis will be the movement of particles out of the cells. So in here we have endocytosis, so you have a particle, and this particle is going to be transported into the cells by these vesicles. Exocytosis will be extruding molecules. So if you magnify this part of the cell, this molecule that was located inside the cell will fuse to this plasma membrane, and then the contents will go out of the cell. So this is exocytosis. Now let's talk about details of the cytoplasm. So the cytoplasm is the cellular material between the nucleus and the plasma membrane. The cytoplasm is made of a fluid portion that is known as the cytosol and proteins that are called the cytoskeleton, plus cytoplasmic inclusions and little organs inside the cells that we call the organelles. Now, within the cytosol, the fluid portion, we have ions like sodium, potassium, calcium, magnesium, zinc, etc. And also we have proteins. So the cytosol is a colloid. We have small proteins dissolving water of the cytosol. Now, the cytoskeleton in the cytosol is made of different proteins. So we have tubulin protein that makes hollow tubes that we call microtubules that are going to be important to transport molecules to form an internal scaffold, and also allows the separation of chromosomes during cell division. We have also tiny proteins that we know as microfilaments that are made of actin. These microfilaments or actin filaments help to maintain a shape within little finger-like projections in the cells that we know as microvilli, and also allows some of the cells to contract. Now we have intermediate filaments that they're not as thick as the microtubules, but they are not as thin as the actin filament, and they can provide mechanical strength to the cells. Along with these cytoskeletal proteins, we have also embedded within the cytosol cytoplasmic inclusions that has chemicals such as fat molecules or lipid droplets and melanin. So this is the characteristics of the cytoskeletal proteins. So all of these that you see here, these are the microtubules. Microtubules are composed of tubulin subunits, and they are thick. They have 25 nanometers in thickness. Now, the intermediate filaments, they have a thickness of 10 nanometers, and they're made of protein fibers. Now, the microfilaments or actin are very thin, are made of these actin molecules that are tiny, and they make out of this diameter or thickness 8 nanometers. So these actin subunits are called F-actin for filamentous actin. Now let's talk about the organelles. So the organelles are small specialized structures that have particular functions. Now, depending on the type of cell, it will be, for the most part, what type of organelles you expect to find most abundantly. So, the nucleus is this central organelle in the cell that has deoxyribonucleic acid. The nucleus it is surrounded by the nuclear envelope that it is made by two layers that has in this layer nuclear pores through which molecules can move in and out of the nucleus. It's like a central office that has, of course, doors. And then the DNA, it is floating within the nucleoplasm or embedded within the nucleoplasm. Now, the DNA helps for transmitting genetic information and also help to direct the activity of the cells. The cells will have other type of nucleic acid like RNA or ribonucleic acid that serves as an intermediate molecule to make proteins between the DNA and the cytoplasm. So we have messenger RNA, which brings the information from the DNA into the cytoplasm so you can make particular proteins. We have other type of RNA that is called ribosomal RNA that actually is in charge of making the proteins. And we have the transfer RNA that is bringing the amino acids necessary to make these proteins. Within the nucleus, we have the nucleolus, that it is a dense region within the nucleus where you make the ribosomes. So this is the structural characteristics of the nucleus. So the nucleus has this membrane. So this is an scanning electron micrograph with a 50,000 magnification of the of the nuclear membrane to show you this double layer with these nuclear pores that you can see as well in here. And you can see them in here. And then in here, we have the nucleolus, which is this. This is a transmission electron micrograph with a 20,000 magnification. And then embedded within the nucleoplasm, which is this, you have the chromosomes. The chromosomes, they have a particular structure. So the chromosomes are DNA that it is coiled into proteins that we know as histones. And these histones, they have subunits, which are known as histone proteins. And this DNA, which is this double helix, will wrap around, like in the old cords of phones, we will wrap around these histone proteins to make the DNA compact into what we call chromatids. And these two chromatids are joined by the central part of the chromosome that is called the centromere. And the chromosomes, they have arms that we call short arms and long arms, and they have endings that we know as telomeres. Now, the DNA, of course, has genetic information in terms of base pairs. So we have adenine and thymine that binds together, and then we have guanine and cytosine that binds together. And then during the cell cycle, which is basically the stage of the cells that is subdivided into periods, within the cell cycle, we have the chromosomes dispersed in the nucleus as chromatin. And during cell division, this chromatin will be condensed to form these chromosomes. The ribosomes will be the protein factory of the cells, and it is composed of a large and a small subunit. The ribosomes is what we call the ADS in animal cells, the ones that makes these proteins. And we have ribosomal RNA, which is abbreviated as rRNA, and this is going to be important to make the proteins. Now we have two types of ribosomes, the ones that are free-floating within the cytosol that are called free ribosomes, or the ones that are attached to one organelle that we call the endoplasmic reticulum. The endoplasmic reticulum is a series of sacs or membranes that are making this network through which the proteins will be made. In the case of rough endoplasmic reticulum, because it had embedded within this membrane these ribosomes. And as these proteins are made, they will be transported into another organelle so that they can be distributed into their particular destiny. Now, the rough endoplasmic reticulum, since it has these ribosomes attached to them, it can be seen in this transmission electron micrograph as series of stacks with a rough appearance. That's why it's called rough endoplasmic reticulum. Now, the smooth endoplasmic reticulum, which will be this, it doesn't have any attached ribosomes. And that's why in this transmission electron micrograph, it doesn't show rough appearance. Now, the smooth endoplasmic reticulum, the function will be to detoxify the cells from drugs and alcohol, and also to manufacture lipids, like it will help us to make the plasma membrane. The Golgi apparatus will be this sac of membranes that helps to pack and distribute the proteins into their corresponding area where they're supposed to go. So this Golgi apparatus or Golgi complex will receive these vesicles that we know as transport vesicles from the rough endoplasmic reticulum, and these will be filled with proteins that will reach into the cisternae, the spaces within the Golgi apparatus, so that they can be transported through these series of stacks, and they can be tagged to go into a specific location within the cells. So it's similarly as to a UPS. It will receive packages and then it will deliver them. And this is the transmission electron micrograph view of the Golgi apparatus or complex and the secretory vesicles that ultimately, in some cases, will be extruded or exported from the cells. Now we have also minor organelles like lysosomes, peroxisomes, and proteasomes. Lysosomes are formed by the Golgi apparatus, and they have enzymes that are going to digest the cell material or the cell itself when the cells are damaged. In this case, you call this autophagy. Peroxisomes will be smaller than lysosomes, and they have enzymes like peroxidase that is going to help to break down the hydrogen peroxide into water and oxygen. Also, the peroxisomes can have this function of breaking down fatty acids and amino acids. Proteasomes are large protein complexes that helps to recycle protein components when the proteins they have a defect. So this is the action of the lysosome. So if this plasma membrane get in contact with certain materials, is going to start to form a vesicle, and this vesicle is going to pinch off from the plasma membrane, and then it will form these vesicles that can be taken into the cells, and then the lysosomes, if this material is toxic to the cells, will come and fuse with these vesicles, and then this material will be destroyed and ultimately will be exported because you don't want to keep those wastes. So as I told you before, the mitochondria is the powerhouse of the cell. It will make ATP. The mitochondria has a sausage-like shape, and it has a double membrane. It has the outer and the inner membrane. The inner membrane folds in to form this structure that you see here and here in this transmission electron micrograph that we call the cristae. And within this inner membrane, you will have this complex where you make ATP through what we call the electron transport chain. The mitochondria has its own DNA, and it has 70S ribosomes, which are similar to the ones that some microorganisms have, like bacteria. Now, where can you expect to see more mitochondria in cells in our body? Well, will be for those tissues who are very active, like in the case of ourselves, the muscle tissue. It's supposed to have more mitochondria because you can generate more energy. Centrioles are located in the centrosome, which is a specialized zone near to the nucleus, like in here. They're made of microtubules in what we call the 5 to 9 rearrangement. And then these microtubules will connect to the spindle fibers in the mitotic apparatus to separate the chromosomes during cell division. Other appendages of the cells that we have are the cilia and the flagella. So they help to propel the cells in the case of flagella. In the case of the cells of the body, only sperm cells have flagella. But in the case of these cells that they have these cilia will be important to move materials on the surface of the cell. So for instance, in the trachea or in the respiratory tract, we have this cilia that helps to move mucus. And this is the way that this cilia looks like. And some of these help to transport molecules. And this is the transmission electromicrograph of these cilia. Microvilli are finger-like projections from the plasma membrane that helps to increase the surface area to absorb substances. And typically the cells have many cilia. Like in this case, this is a transmission electromicrograph of a highly ciliated cells. Inside the microvilli of certain cells, you have lacteals, like in the case of the intestinal cells, to absorb nutrients, like lipids. Others, they have these cytoskeletal proteins like actin to maintain them erect. So those are all the details about the organelles. Now let's talk about genes and gene expression. So the DNA of the cells, they have the genetic information or the genes that are necessary to produce proteins. In order to produce proteins, you have to make messenger RNA. And you make messenger RNA by transcribing the DNA into messenger RNA. Once you transcribe your DNA, you will have the messenger RNA leaving the cells through these nuclear pores. And then, once within the cytoplasm, this messenger RNA can be translated into proteins by adding amino acids one at a time by the transfer RNAs that can read the code of what amino acids come next. And in translation, the information that it is found in the messenger RNA, it will be used to determine the types of proteins that you will make by rearranging these amino acids in the polypeptide chain. So this is the figure to show you how the cells make proteins. So you have your DNA in the nucleus that is going to be transcribed into messenger RNA. The messenger RNA, which is this, is going to exit the nucleus through these nuclear pores, and then is going to reach either the rough endoplasmic reticulum or the free-floating ribosomes within the cytosol of the cell, and then you will have these ribosomes elongating a nascent protein. In this case, will be this. And you will have this tRNA that bringing these specific amino acids to elongate this protein. So how can you transcribe DNA? So you will unwind the two strands of DNA and you will use enzymes that are called RNA polymerases, DNA-dependent RNA polymerases that are going to attach into these nucleotides to make a nascent strand of messenger RNA and always will be following the rules of base pairing, A with T's, C's with G's. Now, in the case of the messenger RNA, you have uracils instead of thymine. And then you will stop transcribing when you have a sequence within this DNA that tells to the protein to not be made anymore. Now, once you have then the cytosol with messenger RNA, the messenger RNA can either go to the ribosomes in the rough endoplasmic reticulum or the free ribosomes to make these polypeptides. So translation involves the cooperation between the three RNAs, ribosomal, then the transfer, and then the messenger. So the transfer RNA will have an ending that is called the anticodon, that is going to bind to the codon of the messenger RNA, and it's going to bring a specific amino acids, like in this case will be aspartic acid. And then you will form these peptide bonds between these amino acids and the nascent tRNA. So again, this is just to show you what happens. So here you have in purple color the ribosomes, and then here you have the tRNA reading this message and at the same time bringing these spheres of amino acids. Or well, they're not spheres, but this represents these amino acids. And then you keep adding them like if it was beads on a string, and then you elongate your proteins. Now the cells, they need to have specific activities at a specific time. And this is controlled by what we call gene expression. So you will turn on certain genes by having signals. So for instance, if a cell needs to grow, it will receive a growth factor, and then you have a signal going into the nucleus so you can increase the transcription of proteins, like in this case, let's say it's a muscle cell, so proteins that make the muscle cells, like actin and myosin. Now, during the lifetime of a cell, the production of protein varies depending as well on the environmental characteristics. And examples will be, for instance, thyroxine from the thyroid that causes the cells to increase their metabolic rate, and more thyroxine will result in having a higher metabolic rate than when someone has less thyroxine. So this is what most of the time we call hypothyroidism when you have lower metabolic rate. Now this is the cell cycle and its different stages. So here we have interphase, and then here we have mitosis. So most of the time, the cells are not dividing, but when they divide, they undergo mitosis or M phase. And then some cells that they don't like to divide, they will enter into what we call the G0 phase. And mitosis, it will be subdivided into prophase, metaphase, anaphase, and telophase. And cytokinesis will be the division of the cell cytoplasm. During interphase of the cell cycle, you will make another copy of DNA. Then once this happens, you have something that's called semi-conservative replication, in which the DNA polymerase will synthesize as many of these bonds you need to make to make a polymer of DNA. However, you have limitations in terms of space, and you will have to pack your DNA into Okazaki fragments that are made by this DNA ligase enzyme. Now, the replication of the DNA happens semi-conservatively, meaning that you have an old strand of DNA, and then you will replicate this to form a new DNA molecule that has this complementary base pairing from this side and similar to the other side. So you form two identical DNA molecules. So what are the specific events in cell division? So in interphase, you replicate your DNA, and in mitosis, you have in prophase the disintegration of the nuclear envelope, so you can have these chromosomes ultimately separated into two new daughter cells. And in prophase, you have the chromatin being condensed to form visible chromosomes. And then you will have then in metaphase these chromosomes aligning into the midline of the nucleus or the cell into what we call the equator. And then these microtubules from the mitotic spindle that are forming from the centrioles into the kinetochore of the chromosomes, they will separate these two chromosomes that are joined, or the sister chromatids into individual chromosomes. Anaphase is this starting point where the microtubules start separating the chromatids, but they're not so far apart. That happens in telophase. And in telophase, at the end, your chromosomes will decondense, and then you can form again your nuclear membrane that is going to protect the newly formed chromosomes. And then you have cytokinesis, in which you divide the nucleus into what you divide the cytoplasm. So this is the structure of mitotic chromosomes. So this mitotic chromosome will convert this chromatin from interphase into two co-joined homologous pairs or chromatids by this centromere. So this is then the diagram of the mitosis and cytokinesis cycles along with the S phase. So during S phase, you have this nucleus and this plasma membrane. And the nucleus can be seen visible in here, but this will be chromatin. For prophase, which is the next phase of mitosis, you have condensing of these chromosomes and disappearance of this nuclear membrane. Metaphase, the chromosomes will align in the middle of the equator, and then you will have attachment of these spindle fibers or mitotic spindle into these kinetochores. And then you will separate them little by little in anaphase and then telophase until you have two daughter cells with identical genetic information. Now, some of the changes that happens with the cells is that they age. And these cells, they age because they have either a cellular clock, they might have dead genes, or they might have damage. Or they can be exposed to free radicals, not radials. And some of the damage that it can happen in the mitochondria. So once these cells get mitochondrial damage, they undergo all of these phases to repair and to form two new daughter cells. So this is all for this chapter. I hope that you enjoy it, and I will see you in the next.